The Krebs cycle is proposed by Hans Adolf Krebs in 1937. It is an important metabolic pathway for energy conversion in the body. It generally involves the oxidation of acetyl coenzyme A to carbon dioxide and water. It is also called as Tricarboxylic Acid or TCA cycle because at the outset of the cycle, tricarboxylic acids like citrate, cis-aconitase and isocitrate participate. It not only supplies energy but also provides intermediates for the synthesis of amino acids, glucose, heme, etc. This cycle connects almost all the metabolic pathways inside the body directly or indirectly. Hence, it is a very important cycle or pathway. The citric acid cycle takes place in the mitochondrial matrix, which is in the close proximity to the location of electron transport system. This is the longitudinal section of mitochondria. The outer one is the outer membrane. The inner one or folded part is the inner membrane. Inside the inner membrane or inner mitochondrial membrane lies the mitochondrial matrix. The electron transport system takes place inside the inner membrane of the mitochondria, and mitochondrial matrix carries out the Krebs cycle or TCA cycle. So, ATP production takes place or inside the mitochondria takes place without any hindrance. Krebs cycle is a cyclic process. Although it is cyclic, many compound enters and leave the cycle, so it is not a closed cycle. Before entering into the cycle or Krebs pathway, the pyruvic acid having 3 carbon is decarboxylated to acetyl coenzyme A in the presence of oxygen. The oxidative decarboxylation of pyruvic acid is the connecting link between the glycolytic pathway and the Krebs cycle or citric acid cycle. The first step includes formation of citrate. Isomerization of citrate to isocitrate takes place in 2 steps, step number 2 and 3. Next, alpha ketoglutarate is formed in step number 4 and 5. Conversion of alpha ketoglutarate to succinyl coenzyme A takes place in step number 6. Succinate is formed in the next step. Succinate to fumarate in the step number 8. Formation of malate, step number 9. And oxaloacetate is formed from malate in the step number 10. And the cycle continues. The first step is formation of citrate. By the action of the enzyme citrate synthase, acetyl coenzyme A and oxaloacetate condense and citrate is formed. The reaction is as follows. In the next 2 steps, isomerization of citrate takes place. The enzyme aconitase catalyzes the isomerization of citrate. In the next step, the dehydration of citrate takes place and cis-aconitase is formed, which is an intermediate product. So, one water molecule is removed. In the following step, hydration of cis-aconitase takes place and isocitrate is formed. That means addition of water molecule takes place. Since these reactions are reversible reactions, so they can operate in both the directions. The next step is formation of alpha ketoglutarate. The enzyme isocitrate dehydrogenase or ICD catalyzes 2 consecutive reactions. In the next step, decarboxylation of isocitrate forms oxalosuccinate. isocitrate is decarboxylated, and electrons are removed and accepted by NAD plus, forming NADH, and oxalosuccinate is formed. In the next step, decarboxylation of oxalosuccinate takes place and alpha ketoglutarate is formed. So here carbon dioxide is removed and alpha ketoglutarate having 5 carbon in its skeleton is formed. See, oxalosuccinate is having 6 carbon, 1 carbon is removed as carbon dioxide, and alpha ketoglutarate with 5 carbon is formed. The next reaction is catalyzed by the enzyme alpha ketoglutarate dehydrogenase. This enzyme catalyzes the oxidative decarboxylation of alpha ketoglutarate. It is dependent on 5 cofactors, namely TPP, lipoamide, NAD+, FAD, and coenzyme A. At this stage, second NADH is produced by removing 2 more electrons and second carbon dioxide molecule is liberated. So, see, alpha ketoglutarate dehydrogenase, second NADH is produced and succinyl coenzyme A is formed. In the next step, succinate is formed. The succinyl coenzyme A is converted to succinate by the action of the enzyme succinate thiokinase. This reaction is coupled with the phosphorylation of GDP to GTP. The GTP so produced is converted to adenosine triphosphate or ATP by the enzyme nucleoside diphosphate kinase. See, succinyl coenzyme A, by the action of succinate, inorganic phosphate is added and coenzyme A is removed and GTP is converted to GTP, and as a result succinate is formed. In the next step, succinate is oxidized to fumarate by the action of the enzyme succinate dehydrogenase. Oxidized means removal of hydrogen. FADH2 is produced in this reaction. That means the removed hydrogens or electrons are accepted by FAD. FADH2 is produced and fumarate results. Then water molecule is added to fumarate and malate is formed. Here the enzyme catalyzing the reaction is called fumarase. Fumarate, by the action of fumarase, water molecule is added and L-malate is formed. See, the reaction is reversible, hence water molecule is added and L-malate is formed, and if L-malate removes one water molecule, then fumarate is formed in the reversible direction. The last and the most important step of the Krebs cycle is regeneration of oxaloacetate. Malate is then oxidized by the action of malate dehydrogenase and oxaloacetate is regenerated. The oxaloacetate molecule can now combine with another molecule of acetyl coenzyme A and the Krebs cycle continues. The last NADH is produced in this step. Hence, in one cycle of Krebs cycle or the Krebs pathway, 3 NADH molecules are formed. L-malate, malate dehydrogenase enzyme, NADH molecule is produced by accepting electrons and oxaloacetate is regenerated. some important points. The TCA cycle basically involves the oxidation of acetyl coenzyme A to carbon dioxide with simultaneous regeneration of oxaloacetate. There is no direct participation of oxygen in the TCA cycle. Still, it operates strictly under aerobic conditions. It is because the reducing equivalents, the NAD plus and FAD required for the operation of the cycle can only be regenerated in the respiratory chain in the presence of oxygen only. The oxidation of acetyl coenzyme A through citric acid cycle produces 3 molecules of NADH and 1 molecule of FADH2. Oxidation of reducing equivalents by electron transport chain coupled with oxidative phosphorylation results in the synthesis of ATP. We will discuss about the electron transport system in some other video. Let's now find out how many ATPs are produced per the reducing equivalents, the NADH and FADH2. There are two rules. According to the old rule, per NADH, 3 ATP molecules are produced and FADH2 molecule produces 2 ATP molecules. According to the new rule, 2.5 ATP molecules are produced per NADH and 1.5 ATPs are produced per FADH2. In my next video, we will discuss about the electron transport system. Thank you for watching.